drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. The full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomes, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode.
I'm a professor at the Jackson Laboratory. I've been here for more than 25 years now. I love living in this part of the world. I was born in Maine, and I live right next to a national park, and there's a lot of benefits to it. I got my degree in mathematics. I went on to get a PhD in statistics, and I've always been interested in genetics. And I came to the Jackson Lab by circumstances of life and really became immersed in mouse genetics. Mm-hmm. So if I had to pigeonhole myself, I would say that I'm a mouse geneticist. And for the past 15 or so years, I've been focusing on metabolism and aging as my areas of interest, but I still retain that mathematics, statistics, data-oriented mindset. So a lot of my work is related to experimental design and interpretation of data.
Yeah. And, many people might not know this, but it's interesting that you have the statistics background and then you went into the realm of biology and genetics. Genetics as a field, it's obviously a field of biology, but it has a very rich history of a lot of hardcore statistics. And I think some people might not realize like how hardcore the stats are that a lot of people, the chops and stats people have when they do genetics.
Genetics is a really quantitative field. I remember as an undergraduate acing the genetics course, and all the biology majors were dreading it. So it's wonderful. It suits me very well. It lends itself to analytical thinking, but it also throws some real biology at you. Biology is complicated and messy. And I will say before we get into it that one of the challenges of dealing with this, unraveling this story, is that it's complex. Mm-hmm. And of course, you want to tell a simple story so that, you communicate something, that it's effective. But I think the complexity is real. Yeah. And I kind of relish it. So,
yeah. And we'll get into that. It's going to be reflected a lot, I think, in this paper that we're going to discuss, just because and this is often some of the most interesting stuff where, I think you have results here that are counter to what most people would have predicted and defy obvious explanation. And we'll talk about some of that. For background, many people might not know what the Jackson Laboratory is, but it's a very sort of important node in the research world. So what is Jackson Labs?
Of course, the Jackson Laboratory is a nonprofit research institute. We're located on the Mount Desert Island off the coast of Maine, founded in 1929 by, moguls who came here for the summer and thought it'd be cool to start a scientific institute. They did. We're most well known for mice. Most of the researchers here work with mice as a model organism. Our focus is on using mouse models and genetic tools to investigate human health, really. That's in a nutshell.
hmm And so, you've got this new paper out that we're going to discuss. That's really interesting. It was quite a massive undertaking, it looks like, and it has to do with the relationship between dietary restriction and health and lifespan. So this is a mouse study. We're going to unpack it. You used a lot of animals here, and you used specific types of mice, and we're going to get into all of that. I want to give people a general base here, some background on some terminology, some concepts, and some limitations and methodological issues that are often inherent to this kind of work that, at least to some extent, you guys are getting around with this study. One of the things I want to talk about here is just mouse genetics at a basic level. So most experiments that are done in laboratory animals like mice are often done in inbred mouse strains. Can you talk about why that is and the types of mice you used in this study and why?
Okay, inbred mouse strains like the C57 Black 6J mouse that was developed here at the Jackson Lab were developed to ensure that experimental results would be more reproducible.
Mm-hmm.
An inbred mouse is created by a long series of generations of brother-sister mating so that uh, each of these mice inherits half of their genome from their mother and half of their genome from their father. And after many generations, those two genomes become more and more identical so that in a sense, they're almost identical twins of themselves,
Mm-hmm.
and they can reproduce exactly the same genotype over and over again. So when we study an inbred mouse, it's really akin to studying one person. And they're also inbred, which is a little unusual in nature.
Mm-hmm.
There are some challenges that come with being inbred, and I know where we're going with this. So,
Yeah, yeah. So on the one hand, having this relatively genetically homogeneous inbred mouse strain or laboratory strain of animals, that can be a feature with respect to research because it can give you more reproducibility. There's less genetic variability that's going to introduce variation and noise potentially into the results. On the other hand, as you say, it might be problematic from another angle to think about this because inbreeding is not something you see in nature, and it's not necessarily going to we might find some things that reflect not what we would see in nature in a more genetically diverse population that might inhibit our ability to generalize results from these particularly bred laboratory strains to other species, including even ourselves.
I think the key word there is generalizability.
Mm-hmm.
If we study C57 Black 6 mouse, at the end of the day, we're really limited in the scope of what we can say to just that particular mouse strain. And anything that we say about other mouse strains or other species like humans is extrapolation, and it may be true, but it's guesswork. So by working with the outbred mice, which I'll explain if you would, um, we increase the generalizability of our studies to at least more mice.
Mm-hmm. Mm-hmm.
And we feel that that's going to increase our potential to generalize to humans, which is the objective here.
Mm-hmm. And in very simple terms, so what is outbreeding here? Is basically the opposite of inbreeding. So you're creating genetic diversity. Is there anything more we should know about that?
The outbred mice, the particular ones that I worked with in this study, um, were originally 8 different inbred strains that we mated together with one another, and we maintained them again for many generations. But instead of mating brother sister together, we randomized the mating so that just like people, the the mother and father of the next generation are not related to each other. And
Mm-hmm.
that makes the genomes all mixed up, and it makes every mouse individual and unique. And that has some drawbacks because we can't put the same genotype in 2 conditions. It would be fantastic to do this study and have the same genotype on all 5 diets, but that's we'll do that.
Mm-hmm.
We'll figure out how to do that. But the outbred mice really, I think, I don't want to say they're a model for human populations because they're not. In some ways, they're a better tool for genetics, but they have the kind of heterogeneity and diversity that you see in humans. And I
Hmm.
think that's exceptionally important for aging because I think everybody ages in their own way.
Hmm.
So each of these mice is going to age in its own way.
Hmm.
Oh, and by the way, outbred means the two copies of their genome are not identical, and that gives them a feature called hybrid vigor. So they're really healthy,
Hmm.
generally speaking.
Hmm. So yeah, coarse grained level, generally speaking, these mice sort of look and act obviously healthier. Presumably they live longer than the traditionally used inbred mouse strains as well, I would imagine.
Well, surprisingly, no.
Hmm.
Their average lifespan is about the same.
Hmm.
It's about 20 months, so a little shy of 2 years average. But what's different is the variance in the lifespan. There are outbred mice can an inbred mouse will have a lifespan that's pretty within a window. There's a lot of randomness to it. And they almost always die of the same causes.
Hmm.
Like a C57 will almost always die of a lymphoma.
Hmm.
They have their own built-in genetic time bomb, so to speak.
Hmm.
And the diversity mice, well, they display all kinds of lifespans and different causes of death. And we've had outbred mice live as long as 4 years and 9 months, which I'm going to say it in dog years is 140.
Hmm.
It's a bit of
I
a
see.
joke.
So I think what you're saying is the average lifespan of these outbred mice is about the same as the inbred mice, but there's more variability in terms of how long they live population wide, more variability in terms of the ways in which they succumb.
Yeah, I could have said that. Thanks.
So basic concepts here. We're talking about dietary restriction and its impact on health and longevity in these outbred mice. We've got calorie restriction, we've got dietary restriction, and then we've got intermittent fasting. What are the essential differences between these three things?
Well, yeah, the language we try to be consistent within our paper, but it's not always consistent across all the literature. So what I mean here by caloric restriction is we fed the mice a standard chow diet that's a healthy diet. We figured out how much a normal adult mouse would eat, and we reduced the amount of food that we provide them by either 20% or 40%. So they're getting 80% of the food they would normally eat or 60% of the food they would normally eat. So those are the caloric restriction.
Mm-hmm.
And intermittent fasting, there's a lot of ways to do intermittent fasting. Probably most of your listeners are going to be familiar with the 16-8 paradigm. It's very popular these days. People restrict their eating to 8 hours a day.
Mm-hmm.
We did something a little different here. We did intermittent fasting where we removed the food from the mice on Wednesday and we gave it back to them on Thursday or Friday. So they either went for 24 hours or for 48 hours without food.
Mm-hmm.
And I know people listening are thinking, well, I could do that. I could go 24 hours without eating, but you're not a mouse. Um,
Yeah.
and one of the things I want to emphasize, and one of my concerns about publishing this paper is that people will look at these diets and they'll think, I should do that. And I would highly advise no. Um, you will not tolerate 40% caloric restriction.
Mm-hmm.
People barely tolerate 20% caloric restriction.
Mm-hmm.
When we fast the mice for 1 day or 2 days, it's a little hard to say what the equivalence is, but it would be like you fasting for 2 weeks.
Yeah, yeah, because they're
And
small animals, their metabolic rate is different, and they're
you wouldn't want to do that.
yeah,
Exactly.
yeah.
So my point is these interventions, CR and IF, as we abbreviate them, they're pretty extreme.
Mm-hmm.
And we needed a term to refer to them both at the same time so that we didn't have to write CR and IF every time
Yeah.
we
So
needed
these are
to
all forms
refer
of
to both.
dietary restriction.
So they're
They're
both dietary restriction, and I use the word dietary restriction very broadly to refer to almost any kind of restrictive
mm-hmm.
intake,
Mm-hmm.
whether
Yeah.
it's timing or calories.
Yeah.
Yep.
And obviously everyone is inherently interested in health and in longevity. But I think the
Yes.
point you're trying to make here is we have to be very cautious about jumping to conclusions about humans here because, 1 day of fasting in a mouse is not equivalent to 1 day of fasting in a human. We're talking about fairly strong interventions here. And I think that would imply that all of the results we're going to talk about are right. We need to keep in mind that these are strong interventions. And so any more mild forms of fast. So if I fast for 24 hours here, I should expect, if anything, weaker effects than what you saw in the mice, for example.
I would not argue with that. And I would also say that there can be benefits to reducing calories and to fasting.
Mm-hmm.
We just need to be a little careful. And what we're trying to do with the mice here is to push the envelope because we're interested in the fundamental biology. Ultimately, we want to understand the mechanisms. And one possible outcome of these studies down the road would be to understand the mechanisms and find different ways to intervene so that we could use our understanding derived from calorie restriction to find other routes to improve health.
Mm-hmm. And so I want to give people a basic sense before jumping into the results themselves of where this field, broadly speaking, sort of stood and what some of the motivation to do this work was. So obviously, people have been talking about calorie restriction and dietary restrictions of different kinds for some time. Intermittent fasting is very popular. And I think sort of a headline summary of this field is that in general, people often find that dietary restrictions like this tend to result in health benefits and increased lifespan. Is that basically what the thinking has been, and how does this tie into what is known and unknown about what was actually causing these lifespan benefits from restrictions?
Okay, it's a little nuanced, but
Mm-hmm.
let's start with um, the dietary restrictions. Caloric restriction in particular has been known to extend lifespan in rodents for
Mm-hmm.
100 years.
Yeah.
Um, we We also have seen research in the past decade or two suggesting that not every genotype, so remember, if we look at inbred mice, if we calorie restrict C57 Black 6, they live longer. But it wasn't really clear that all mice would benefit from caloric restriction. And I think that was the biggest motivator. Well, I did this. It was the biggest motivator for doing this study without bred mice to ask the question whether caloric restriction would be universally beneficial or
Mm-hmm.
whether there might be subsets of animals that really didn't benefit from it in terms of
Yeah.
lifespan.
Yeah.
Lifespan is really is is a measure of aging, and I would like to, if you would help us come back to that point
Mm-hmm.
in a bit. The question I would ask though is, do you want to live as long as you possibly can, or do you want to be healthy while you're alive?
Yeah. Health
And,
span as opposed to lifespan.
so we did focus on lifespan as our primary singular outcome, but as you know from looking at the study, we measured everything under the sun related to health,
Yeah.
everything
Yeah.
you could think of. And in particular, these interventions improve metabolic health. And this is a really good thing for people because we have such a high prevalence of metabolic disease. And we saw these improvements in metabolic health in the mice on the diets.
Mm-hmm.
So extend lifespan, improve health with some caveats.
Mm-hmm. So another thing that you did here is not only are you using outbred mice, they're more genetically diverse. You used a lot of mice. This study involved almost 1,000 mice. Obviously, this gives you statistical power. You also needed to do that more just sort of by necessity because you don't have genetic homogeneity. So there's going to be more natural variation from mouse to mouse. You're looking at, 5 different cohorts of mice that get to eat whatever they want all the time. You've got mice that have, 1 day of fasting imposed on them, mice who have 2 days of fasting imposed on them. And then you've got the 20% and 40% restricted groups. They can eat as much as they want whenever they want, but there's this limit on the total available calories to them. And as you said, just keep in mind, everyone, we're not going to list every single thing that you guys measured, but you measured a lot. Can you give us just a basic sense for the diversity of measurements and biomarkers and things?
I will talk about the scale that the mice from the first cage that we filled until the last mice died spanned almost 8 years.
Wow.
At the peak of the study, we had 7 full-time technicians just managing the phenotyping schedule. Um, we really learned a lot. We did all kinds of clinical assays. We weighed them every week. We put them in an NMR, an imaging machine to look at their body composition. We put them in metabolic cages for a week at a time to see their circadian rhythms and their metabolism, their activity. They went on wheels. We did multiple blood draws. They got echocardiogram. I'll stop. You get the idea.
Yeah, yeah.
Um,
Everything, like everything under the sun.
yeah, why not? You're just going to do this once, you
Yeah,
know?
yeah. So very thorough, very, very massive study. 8 years of time, it sounds like. One last piece to mention maybe is I think you looked at female mice only. Is that just to decrease? Is that true? And was that just to sort of
We
decrease
did.
the?
We looked only at female mice,
Mhm.
and I think that's a weakness of the study. But I'm going to add that there have been so many studies that have been done only on male mice that I feel like it was fair to tip the teeter-totter the other way.
Mhm.
Um, One of the issues with the diversity mice is that they are genuinely real wild mice.
Yeah.
And in the wild, female mice tend to live together rather well, and male mice not so well always.
Mhm.
It doesn't happen all the time, but if you leave male mice together for a long time in a cage and you deprive them of food, they fight.
Yeah.
And once the mice start fighting, they get injured and you're obliged to euthanize them.
Yeah. Yeah.
And, we
I s-
felt that,
but anyway, so you've got almost 1,000 female mice in this study. What's the first basic result here? So you've got these five different groups of mice that are on five different feeding schedules, basically. What's the headline result here in terms of how raw lifespan differed between the groups?
well, the basic result is that these dietary interventions all extended lifespan. And you might say to yourself, that's not a surprise, but I feel like it was somewhat surprising to me, especially with the 2-day intermittent and the 40% caloric restriction,
Hmm.
because I felt that one of the likely outcomes is that these interventions would have such severe detriments that we would shorten the lifespan of some animals.
Hmm.
And honestly, I didn't see that. There was no indication that there was any fatalities due to the interventions. So that was a good outcome for us. But in the strictest sense, as a statistician, I think what we've demonstrated here is that when we assign these diets at random, they extend the lifespan. And because of that, we have a causal relationship. Dietary intervention causes lifespan extension.
Hmm.
And I'm looking at my notes here because I want to get this right. But yeah, I want you to think of a little circle with diet written in it, and a little circle over here with lifespan written in it, and an arrow that points from diet to lifespan with a plus sign on it.
Hmm.
And we established that. But why do we measure all that other stuff? I'm sorry, I'm not supposed to ask you questions,
Well, I
but
mean, so I mean, there's some natural questions here. Is yes, these mice may be living longer, but are they more sickly? Are they more frail? Do they have less energy? Are they less healthy in some sense based on other measures we might care about?
what we saw, and when you let's talk about the 40% CR mice because they are so extreme. All the mice on interventions lost weight.
Mm-hmm.
The 40% CR mice lost weight continually through their lifetime. They lost fat mass, they lost lean mass.
Hmm.
This is not good.
Yeah.
You would not like that. But when we look at the mice, we handle them. They're really quite healthy.
Hmm.
They don't have any obvious deficits. They're not sick. In fact, we know that we reduce the incidence of palpable tumors, so they're likely not getting cancers while they're on these interventions. Um, their blood glucose stabilizes. They don't get obese. I could go on and on, But when we looked a little deeper, especially when we looked at properties of the immune system and we looked at the red blood cells, there were definitely indicators that these interventions um, are not universally good.
Hmm.
And I think, whenever you manipulate a complex system in any way, you're going to have good and bad, right?
Yeah. And maybe something important to keep in mind as we go through some of the details is, the mice in these groups did have lifespan effects. So 40% calorie restriction resulted in quite a substantial increase in lifespan. But as you said, and as we'll keep in mind as we go through some of these biomarkers and other things, um, I want to emphasize too that these mice are living in assisted living for their entire lives. They don't
Oh,
have to worry
yeah.
about dense packing in large groups that might result in pathogen transmission. They don't have to worry about getting eaten by an owl or a coyote or a predator. And so I would imagine that some of the things that we talk about, even though these mice live longer with these dietary restrictions, such mice under such conditions, if sort of transposed into the wild, might actually live shorter lives for various reasons we can imagine.
This is the generalizability question.
Mm-hmm.
I can say that under these conditions, the dietary restriction extends lifespan. You were right. There are no predators. There are no pathogens. They live in a clean facility. We do house them in large groups because they're
Mm-hmm.
social. They like to be, not packed in, but they
Mm-hmm.
like to live close to one another. Um, yeah, they're healthy mice.
Mm-hmm.
And they're healthy mice because they get a good diet and we take care of them. Veterinarians look at them every month. And yeah,
Mm-hmm. So getting into some of the results here, yeah, can you give us just a little bit more detail on how some basic measures changed across the lifespan in the different groups, body weight and body composition, adiposity, things like that?
yeah, we saw some interesting differences between caloric restriction and intermittent fasting. And the one that really struck me is that mice who are feeding ad lib, so all you can eat, which mice self-regulate, they do a good job. They have healthy diets.
Mm-hmm.
They tend to grow and they gain lean mass almost throughout their entire lifespan.
Mm-hmm.
And the intermittent fasting mice, despite their weekly deprivation, if you would, gain lean mass. They lose some fat mass, and so they're leaner, but they don't lose that muscle. They don't get smaller.
Hmm.
Caloric restriction mice lose everything. They lose lean mass. They get smaller. That's one thing we noticed. Another thing we noticed, and that is, I need to talk about red blood cells for a minute.
Mm-hmm.
You want to give me permission to do that?
Yeah, absolutely. Yeah, yeah, yeah.
Red blood cells are the most common red cell in your body. You've got trillions of these things,
Mm-hmm.
okay? And they not only do they deliver oxygen to your whole system, but they do all kinds of biochemical things while they're flowing around through your circulation. And a red blood cell lives about 120 days in circulation. And the way that happens is as red blood cells age, they tend to get bigger. And your spleen is constantly monitoring the red cells and it'll identify the big ones and filter them out. So it's keeping the red cell population healthy.
Mm-hmm.
So one of the measures that we take on these mice is we look at their red cells. We know how big they are, we know how much hemoglobin they have in them, and we know their distribution. We know how big they are. And what happens in especially the 2-day intermittent fasting, but also in the 40% CR mice, is that distribution gets wider. And that tells us that the spleen is not doing its job efficiently. It's not
Mm.
clearing out the red blood cells the way it should be. And this measure, RDW, it'll be on your on your report from your annual health because it's typical clinical measurement from a complete blood count test,
Mm-hmm. Mm-hmm.
is already known to predict lifespan in humans. And here we're looking at interventions that extend lifespan, but they also increase RDW, which reduces lifespan. And I, and this is a pattern and a theme that comes up over and over again. And remember, diet arrow pointing to lifespan with a plus sign. Now, that arrow has hidden in it all kinds of things, and that's why we measured all kinds of things. We want to pull that arrow apart and find out, well, what are the steps along the way from diet to lifespan? And one of those things is RDW. So you picture an arrow from diet to RDW with a plus sign, but then RDW to lifespan with a minus sign.
Hmm.
It's contradictory.
Right.
And we saw this again and again. The diet makes something worse, but the diet extends lifespan.
Hmm.
Um, we
Yeah.
saw that. Yeah,
And
go on.
I think we'll come back to that, but is is the punchline here that that is just mysterious at this point?
Is it mysterious? No, I don't.
Hmm.
I don't think it's mysterious at all. I think that the diets don't have a singular effect.
Hmm.
They do lots of things to the system as a whole. And if you think about what lifespan means, lifespan is how long the animal lives. Animals are going to live for a period of time that is to a large extent random, but it's also determined by many, many things. And they die of the first thing that kills them.
Hmm.
And there are many ways to die.
Yeah.
And,
Yeah.
if you have failure related to your red cells, or if you have other kidney decline or cancer, so diet affects lifespan, but lifespan is a complicated thing.
Hmm.
We want to know what are the components of lifespan that are affected by diet. And some of those are affected in a good way, and some of those are
Hmm.
affected in a not so good way.
I see. So for simplicity, if we think of our arrow, it's going from diet to lifespan. Let's just say
So
inside of that arrow, there's actually 100 different factors, 100 different ways,
at
things that
least
could
you got
break
it.
at least.
Yeah.
But for simplicity, let's say 100, and some the dietary restrictions are having some pattern of effect on all or many of those things. And so even if some subset of those things goes down or becomes more negative, it's a bad effect. There might be a boosting or an increase or a positive effect on some greater number of other things, such that the net effect is that as a population, you tend to see a lifespan extension.
That is the key as a population. So let's clarify something we said earlier. When we say the dietary restrictions extend lifespan, does that mean that you as an individual could go on dietary restriction and you're guaranteed to have a longer lifespan? No. It would, on average, increase your chances. But when I say on average, I'm really talking about a population, and in this case, a large group of mice on dietary restriction means, for whatever reason, some of them manage to live longer. Um, so it's not that there's a guarantee that this intervention will extend lifespan, but it's going to do lots of things. And depending on luck of the draw and genetics,
Mm-hmm.
maybe it's going to extend lifespan or not affect lifespan at all.
Mm-hmm. Mm-hmm. I want to talk a little bit about, for a few minutes here about metabolic effects. And obviously, when you restrict an animal's diet, whether it's the calorie restriction by 20 or 40%, whether it's the 1 or 2 days of imposed fasting that some of the other mice got, obviously this is going to have metabolic effects. It's going to influence things like glucose homeostasis, like insulin resistance, like daily energy expenditure. Can you talk about some of those variables, some of those big metabolic variables, things that we knew would change, and how they actually changed across the groups and what the relationship was with lifespan?
Okay. When we talk about metabolic health, um, you have to keep in mind that the situation for modern humans is very different from the situation with mice in a mouse room.
Mm-hmm.
Um, it actually there are a lot of scientists who would contend that caloric restriction works because we cure obesity and all the associated detriments that go along with obesity.
Mm-hmm.
Mice, at least the diversity mice, they don't they don't necessarily get obese. Some of them do, some of
Mm-hmm.
them don't. They're all
Mm-hmm.
different.
So you're just
Um,
saying the mice that had free access to food for their whole lives for the whole duration of this experiment, they gain weight on average throughout their
gain weight,
lifespan, which is normal for animals as they get older and grow and everything. But they weren't becoming obese.
right. So out of 1,000 animals, 160 plus of them on the ad lib diet, we had one overt diabetic mouse with a
Hmm.
blood glucose that went up over 300.
Mhm.
So they're generally healthy, but let's come back to what the metabolic effects are.
Mhm.
So the diet causes a reduction in body weight, which is largely fat, so they're leaner. Um, their glucose levels go down. Um, what else? Their body temperature goes down. I would say that's not a desirable thing. They're cold.
Hmm.
Uh, their activity is interesting because when the mice are on IF and they're not being fed, their activity is low, their energy expenditure is low, but then they refeed and they have high activity. If you go in the mouse room and look at the mice, the restricted mice are the active ones. They're popping up and down and jumping around. And that's because they know you're there and
Mm.
they know where food comes from. So
They're getting excited by the presence of the human that predicts food.
yeah, so even though the impression is that they're more active, they're actually less active.
I see.
Although I will say that 40% CR mice retain their activity level throughout life really consistently.
Hmm.
Did I digress too much? But
No,
the
I mean,
obesity
so,
goes down and the blood sugar stabilizes, and these all sound like really good things, and they
yeah,
they are in a human context, they are good things.
yeah. So you restrict the animal's diet, 20 to 40% of calories restricted, or they're on this 1 or 2 day a week fasting schedule where there's no food access. You're saying you see a lot of expected changes, stabilization of glucose levels, decreases in energy expenditure, decreases in body temperature, things that you would expect to change that go in the expected direction. And yet,
Okay, so this comes down to the statistical details of how we understand the data.
mhm,
But let's suppose that the diets are working by reducing obesity.
yeah,
Then what should happen is if I look at animals within one diet,
yep,
this is stratification.
mhm,
If I just look within one diet, if the reason that diet is extending lifespan is due to reduction in obesity, then the animals that lose the most weight should live the longest.
yes.
That's the expected result. But what we actually see is the animals that lose the most weight live the shortest.
Mhm. So let's say that again. So within a group, so you're restricting the calories that these animals can take in.
Yeah.
So you impose that thing on a group of mice, and they live longer as a population. The thinking is, okay, well, you're reducing calories. Naturally, the mice will be leaner, they're less fat. And so, if that is the reason they are living longer, then within that group, the ones that lose the most fat should tend to live the longest, and the ones that lose the least fat should tend to live the shortest. But what you're saying is that you observe the opposite.
The opposite. Yeah. And
So
this
within a
is,
group, the ones the ones that are the least lean are tending to live longer.
yeah, I would say that these diets, they cause extended lifespan. They cause reduced body weight. But if we look within a group and we ask who lost the most weight, they have shorter lifespans, and that is telling me that the reason for lifespan extension is not lost weight.
Hmm. What about body temperature?
It's the same story for body temperature.
Hmm. And I guess
Um,
naturally, sort of maybe a naive prediction here would be like, okay, if I'm restricting calories, the body has just got less metabolic activity going on. I'm slowing
yeah,
things down. I'm cooling
I'll
things
last
down,
longer.
and therefore the engine will burn hot less often, and I'll live longer.
Mm.
Is that
That
basically how people would think about it?
that is how people have been thinking about it. Yeah.
And then you see the opposite within a group. It's actually the ones that have the higher body temperature that tend to live longer.
Hmm. Yes.
Hmm.
And I'm going to put my interpretation on this.
Yeah,
It is that I think the diets are stressful.
they're stressful.
Yeah. And obviously, just think about it. Someone takes away half your food. This is
Yeah,
stressful.
yeah.
The diets are stressful. Stress has some benefits. Stress has some downsides,
Yeah,
like our,
you want some stress, but not too much.
yeah. So the animals that better tolerate the stress, they lose less weight.
Which
You
makes
know,
sense. Yeah.
yeah. And the animals that are more sensitive to the stress, they're going to lose weight. I think weight loss is a really good indicator of stress in a mouse.
Hmm.
When they're stressed, they're going to lose weight. They burn more energy. And so I think that animals coming into a diet group that are already intrinsically more resilient deal with the stress of the diet better and they
Hmm.
gain the most benefit.
Hmm.
And the animals that come into the diet and they're already maybe not that robust are going to be hit harder by the diet. They're going to lose weight and they're naturally going to have a shorter lifespan.
Hmm.
So,
Did you
uh,
have any way to measure something like stress resilience by other means?
measuring stress resilience is a big open question for the field. You can imagine that there are some ethical considerations. We're very serious about how we handle animals, and
Hmm.
of course, we want to avoid stressing animals. So the way we measured stress resilience was actually after the fact. During their lifespans, I told you how much phenotyping, how much measurement we made on these animals.
Hmm.
There was a period once a year where they spent a whole month in the facility where we did all these procedures, where we weighed them and put them
Hmm.
in the NMR machine and put
Yeah.
them in metabolic
You have to
cages.
pick them up. You have to disturb them. It's
Yeah.
stressful.
And part of that's, I think the biggest stressor for them is to be separated from their cage mates
Hmm.
because
Hmm.
when they're isolated, they're looking for their cage mates. They know something's disrupted. They're not in their home cage. And what we noticed is during that month, almost all mice lost weight.
Mm.
No surprise, it's stressful.
Yep.
So I thought, well, how much weight did they lose? And so we went through and we weigh them all the time. We weigh them at least once a week. So we went through and we blocked off that month from beginning to end and measured how much weight did they lose.
Hmm.
What we found is that the more weight they lost, the shorter their lifespan would be. And this was true within every diet group. And
Ah.
again, like I was saying before, I think this is a measure of how resilient the animals are to stress.
Yeah. So the idea here is what's nice about this is you're finding something that cuts across all of the conditions. So within every condition, the mice might have access to food at all times. They might have 40% of their calories taken away. They might be on this intermittent fasting schedule for 1 or 2 days. Within each group, when you do this month of testing, you could measure how much weight they lost, and they would all lose weight because that's stressful and it just results in weight loss. But you're saying the ones that lost the least weight, which implies that they're probably more stress resilient, tended to live longer no matter what the feeding condition was.
Yeah, isn't that amazing?
Yeah.
I,
Wow.
I
And it
love it.
sort of, it sort of really does kind of start to twist and change a little bit how you think about things like dietary restriction as it's not merely changing the pattern of food that an animal eats. It is a form of stress that must be dealt with by the organism.
Mhm. Agreed. And stressors can have beneficial effects because in you think of a mild stressor is enough to stimulate your body's homeostatic response. Your
Mm-hmm.
body will respond to a stress by bringing compensatory mechanisms on board. And if you stress an animal severely, you're going to do damage to them, and that's not good. So we avoid that. But if you stress them a little bit, they and I can't prove this with the experiment that we did. We're going to do some more. But I think the diets could potentially also trigger these homeostatic responses and induce more resilience than background,
Mm-hmm.
if that
Mm-hmm.
makes sense.
Yeah. And so one of the things that you found as sort of like an overarching thing is like figure 3, for example, it's titled Health and Metabolic Traits Change with Age and Diet, but are poor predictors of lifespan. So you guys measured all sorts of stuff: cardiac output, grip strength, prevalence of tumors, just wheel running activity, all sorts of stuff. And you found that many of these things were changed by diet and did change across the lifespan, which were unsurprising, and yet they poorly predicted lifespan. Can you give us some examples there, and maybe sort of what sort of the overarching result was in your view?
Um, yeah, cardiac output. I forget which way it goes. Let's see if I have actually have figure 3 somewhere.
Um,
Did we look at?
yeah, I've also got it right here.
I can't even read this thing. I pored over every word for so long. I can't even do it. Um, what did we put on there? Oh, fasting glucose, body
Yeah.
temperature, and frailty. Hmm. I lost the question. Sorry,
Okay,
guy.
hold on. Let me pull this up here. So you measured basically you measured a bunch of health and metabolic traits.
We did.
And some of these change with age, as you would expect. Animals get more frail with age. You expect something like grip strength to go down with age, I imagine. But nonetheless, even though they changed as a result of the dietary interventions, even though they changed across the lifespan, they were often poor predictors of lifespan.
Right. Let's go back to the arrows. And if we think of it
Yeah.
as the diet is causing changes in these traits.
Yeah.
So the diet has an arrow that points to cardiac output. It has an arrow that points to grip strength. And it affects these things, but there's no arrow from grip strength to lifespan. There's no arrow
Yeah. Yeah.
from surprisingly cardiac output to lifespan. So the diet has lots of other effects that are not intermediates for lifespan.
Yeah. Yeah.
It's
Yeah, and
hard.
they appear not to be intermediates, even though many people would have expected them to be.
Yes.
Yeah.
And again, improving metabolic health is a good thing, especially for people.
Hmm.
Improving cardiac output, it's a good thing. So it's do you want to live as long as you possibly can, or do you want to be healthy while you're alive? And I
Hmm.
think some of these effects are definitely in the healthy direction. They're beneficial and they're good, and they don't necessarily extend lifespan. So we shouldn't equate good health with long lifespan.
Yeah. Yeah.
That's a punchline.
Hmm. Were there any things that looked like were there any things that were clearly associated with longer lifespan that you would interpret as being like less healthy or less desirable to have if one is alive?
Oh, absolutely, the weight loss.
Yeah.
Uh, yeah, I think that's the big one.
Yeah, just losing all of that mass, including that lean mass.
Yeah.
Yeah, and I mean, so again, we don't want to anthropomorphize, but give us a sense. So remind us again how much. So like in the 20 or 40% calorie restriction group, that's quite a severe restriction. Remind us how much fat and how much lean mass was lost there. Quite a bit.
Oh, that is a good. They were about half the size that
Yeah,
they would be on an ad lib diet.
yeah, yeah.
So
So in a human being, that would be, that's
that
imagine
would
being, I
be
don't know, roughly half as big as you would have been.
yes, and not desirable. Just put
Yeah,
it that
yeah. And do you think so? So things like that in these conditions, we mentioned this before, under these conditions, these things are associated with longer lifespan, but it's very easy to imagine that such a mouse living out in the real world outside of assisted living, those things would actually be a huge liability.
Well, let's talk about the immune system for a minute. And I'm
Mm,
on thin ice here, and I really did rely on my immunologist colleagues at Calico and at the Jackson Laboratory to help us understand what the immune system changes were.
yeah.
And when animals went on these diets, the caloric restriction, one thing we noticed is reduced inflammation. And this is
Hmm.
definitely a good thing.
Yep.
We saw fewer inflammatory macrophages, and we presume based on that we have less inflammation. But there's a whole population of cells that are called NK. They're natural killer cells, and their job is to go sniffing around for viruses, tumor, bacteria, and set off the alarm bells and start the process of attacking the virus or getting rid of the bacteria or addressing the tumor. And these cell populations were badly depleted by the diets. So a mouse in the wild would be exposed to all kinds of pathogens, viruses everywhere. And if that mouse were deprived of calories to this extent, its immune system would not function well
Hmm.
and it would not survive.
Hmm. And is the basic interpretation there simply that is it basically a common sense interpretation? The immune system is important and it's metabolically expensive. There's a lot of fuel that needs to run an immune system to deal with an infection, clear it out. And therefore, if you substantially restrict the diet like this, it's probably not surprising that the immune system is not going to be able to run at full throttle.
I think that's absolutely true. It's accurate. And there's another thing to know about the immune system, and it's a very knife edge thing because when your immune system kicks in and it's fighting an infection, it does a lot of damage. So there's a real fine balancing act
Mm-hmm.
between how reactive your immune system is. We know with COVID, for example, um, when people would die of this, it would often be because of the inflammatory effects that are the sequelae of the immune reaction and, not a direct result of the virus. So your immune system has to be kind of in this just right place.
Mm-hmm. Yeah.
And in the dietary restriction, at least that component of the immune system represented by the NK cells is kind of down.
Mm-hmm.
It's not up to speed, so it won't protect them from viruses. The flip side is they don't have inflammation
Yeah.
because their immune system's not
Mm-hmm.
overreacting to things. It's not,
Yeah. Yeah,
yeah,
so they're probably on the one hand benefiting from lower levels of chronic inflammation. They're less likely to have an immune system that's inflaming the body too much in response to an injury or something like that. On the other hand, they are more susceptible to infection.
that's what I would guess. We didn't
Presumably,
actually
yeah, if
test.
you were to
Yeah,
put them out in the real
yeah.
world.
Everything is context,
Yeah,
and
yeah.
the mice live in SPF, and we keep it pretty safe and clean for them.
Hmm. Um, so there's another sort of major result here that I want to talk about. So it was really cool. You were using so many mice in the study for such a long time. You had these genetically diverse mice. They aren't the inbred mouse strains that are typically used in experiments. So now you can look at actually genetic variability as a variable here. You can see how things vary as a function of the genetic distance between individuals within each of these groups. And there's a result here that I personally do not find very surprising at all, that I think is very important. I think some people maybe will find it surprising. Some people might find it, I don't know, a little upsetting or something sobering maybe. And obviously when we're talking about this stuff, dietary restriction, these are interventions in human beings. We can choose to do things like intermittent fast and calorically restrict and go on diets and things like that. And the whole point is we want to use um, we want to choose to do certain things that are healthy for us, that are beneficial for us. But you've got a result here that speaks to just the role that raw genetics played here. What was the relationship between the genetics of individuals and how long they lived?
Okay, this is a tricky one to navigate. So
Mm-hmm.
let me try and say, when we talk about nature versus nurture,
Mm-hmm.
genetics versus environment, it's always both,
Mm-hmm. Yeah.
and it to different degrees. And these mice are in really controlled environments. The
Mm-hmm.
temperature never changes. They get good food. The same thing happens. There are no pathogens, no predators. You pointed these things out. So um, those constant environmental conditions really raise the impact of the genetic portion of the equation. I would guess, and I don't have to guess because the data are out there, that the heritability, the proportion of lifespan in humans that's attributable to genetics is smaller, much smaller than the 24% that we saw here in the mice. Um, why is the genetic effect of interest to us? we can attribute 24% of the variability in the lifespan to genetic factors.
So almost a quarter, which is quite a lot, right?
Yeah, it's a lot. It's
Yeah.
really it's a whopper. Um, and only I forget the number, but only 7 or 8% to diet.
hmm
Remember, everything's context dependent. So in this context, but the genetics gave me a ruler to measure how big the diet effect is. So remember, if you go on one of these diets, it's not a guarantee that you're going to live longer. It
hmm
improves your odds, so to speak. Um, and how much is it contributing relative to having a 90-year-old grandmother?
hmm
And you really want that 90-year-old grandmother.
Yeah,
There's,
that's what this
there's
result is
nothing,
basically
there's
saying.
nothing you can do about that. You can't choose that.
hmm
Um, this is not, not your fate, though. There are a lot of things you can do within the hand that you're dealt to make yourself healthier and live longer. But it is what it is, and it's an important component
hmm
of. And there's a third component here, and that is everything else that we don't understand.
Yeah.
I'm going to say that's 70% of it.
hmm
I'll call it noise for today, but it's the randomness of lifespan.
Yeah, just like because there's so much in biology that is dependent on stochasticity. Just
The bump,
something happens to go one way versus the other.
the bumps in the road.
Yeah. Yeah. The full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure, and vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.